Hello friends, today I will be discussing about the pyruvate dehydrogenase complex or PDHc. This is a very important enzyme which links the glycolysis to the TCA cycle, or we can say it is the connecting link between the glycolysis and TCA cycle. How? Let's see. So as I told you, it links glycolysis to the TCA cycle. Now we are going to see how. We know the end product of glycolysis is pyruvate, which is a 3-carbon compound, and it is present in the cytosol. And here is the we are going to see that this PDHc is going to convert this pyruvate into the acetyl CoA, which is acetyl CoA is a 2-carbon compound. Now, first this pyruvate is brought in the mitochondria with the help of the transport protein like pyruvate mitochondrial carrier. These are the transport proteins which are present on the inner mitochondrial membrane. So this pyruvate now is present in the mitochondria after the transportation by this transport protein. Here it will be catalyzed by the PDHc, pyruvate dehydrogenase complex. Mainly three steps occur. One is the oxidative decarboxylation, second one is the transfer of the acetyl group to the coenzyme A and the production of NADH, and acetyl CoA is formed, and this acetyl CoA enters the TCA cycle. So it is the connecting link between the glycolysis and the TCA cycle. Now we'll see this pyruvate dehydrogenase complex consists of 3 enzymes, 5 coenzymes and 2 regulatory enzymes. Very important to remember. Now what are these 3 enzymes? First one is the pyruvate dehydrogenase, second one is the dihydrolipoyl transacetylase, and third one is dihydrolipoyl dehydrogenase. So which are represented with E1, E2, E3 respectively. And there are 5 coenzymes associated with them, and each enzyme have a specific coenzyme. Like pyruvate dehydrogenase is associated with the TPP, which is thymine pyrophosphate, which is the derivative of vitamin B1. Then this dihydrolipoyl transacetylase is associated with the 2 coenzymes, that is lipoic acid and the CoA. And dihydrolipoyl dehydrogenase is associated with the coenzyme FAD and NAD positive. Now, these 3 enzymes have a respective role. Like pyruvate dehydrogenase help in the oxidative decarboxylation of pyruvate. Then dihydrolipoyl transacetylase help in the transfer of acetyl group to the CoA. And the function of dihydrolipoyl dehydrogenase is to regenerate the oxidized lipoic acid. So this we have discussed. These are the 5 enzymes, 5 coenzymes, and the vitamin associated with them, or these are the derivative of these vitamin. Like thymine pyrophosphate is the derivative of vitamin B1, flavin adenine dinucleotide or FAD is a derivative of B2, which is riboflavin. Coenzyme or CoA is the derivative of pantothenic acid, which is also represented with the B5. And nicotinamide adenine dinucleotide or NAD is the derivative of vitamin B3, which is also known as niacin. And alpha lipoic acid is not a derivative of vitamin, but it's a coenzyme of dihydrolipoyl transacetylase. Now, the 2 regulatory enzyme associated with PDH complex is the pyruvate dehydrogenase kinase, which is represented with the PDHk, and second one is the pyruvate dehydrogenase phosphatase, which is represented with the PDHp. Now, this is the best image to present the functioning of these 2 regulatory enzymes, that is pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase. So the main step or main concept to remember is that this pyruvate dehydrogenase complex is active when it is dephosphorylated. Dephosphorylated means when phosphate is removed. So here you can see this is the inactive form of the pyruvate dehydrogenase complex, which is consisting of 3 enzymes E1, E2, E3. PDH means pyruvate dehydrogenase, DLAT means dihydrolipoyl acetyl transferase, and DLD is dihydrolipoyl dehydrogenase. So when it is phosphorylated, it is in inactive form. So the pyruvate dehydrogenase phosphatase will remove this phosphate and it will become active. Okay, so the magnesium and calcium, they promote this enzyme activity, so it help in the activation of pyruvate dehydrogenase complex activity. So pyruvate will be converted to this pyruvate dehydrogenase complex by phosphorylating with the help of ATP. It will, you can see now phosphate is added to this pyruvate dehydrogenase complex. Now it will become inactive. So whenever the product is more, acetyl CoA or NADH is more, it will activate this pyruvate dehydrogenase kinase, which will cause the phosphorylation of the this enzyme, and it will become inactive. So acetyl CoA will not be formed. So whenever there is a acetyl CoA to CoA ratio is more or NADH to NAD positive ratio is more, this will activate this pyruvate dehydrogenase kinase. What it do? It phosphorylates this enzyme and make it inactive. Now the mechanism of action of pyruvate dehydrogenase complex. So first, the pyruvate, which is a 3-carbon compound, will be converted to the 2-carbon compound, because we have to form the 2-carbon compound acetyl CoA. Now it will oxidatively decarboxylate, means carbon dioxide is removed. Now this TPP, which is the coenzyme of the first enzyme, that is pyruvate dehydrogenase, will receive this hydroxyethyl group to form hydroxyethyl TPP. This lipomide, which is covalently linked with the E2 or dihydrolipoyl transacetylase, will receive this hydroxyethyl group or acetyl group to form acetyl lipomide, and it will get reduced. Now this acetyl will be received by the CoA to form the acetyl CoA. So the two main step of the pyruvate dehydrogenase complex is complete. Now this reduced lipomide will be reoxidized by the FAD linked dihydrolipoyl dehydrogenase, and this FADH2 will be oxidized back with the help of NAD positive, and NADH is formed, which will enter the ATC chain, which is present on the inner mitochondrial membrane, and it will help in the production of 2.5 ATP. Which is asked in the exam is the enzyme similar to pyruvate dehydrogenase complex. First one is alpha ketoglutarate dehydrogenase complex, and second one is the branch chain alpha ketoacid dehydrogenase. So you can see the first alpha ketoglutarate dehydrogenase is also having the 5 coenzyme thymine pyrophosphate, lipoic acid, and carbon dioxide is removed. Just like pyruvate dehydrogenase complex, FAD is linked, NAD positive is there. So it is having same 5 coenzyme as pyruvate dehydrogenase complex. And second, you can see here alpha ketoacid dehydrogenase.